In this video, we're going to talk about chromatography in terms of paper chromatography for GCSE. Now, what is chromatography? Chromatography is just a separation of colours. So it's a separation of colours using and separation of dyes at this level. It does get a bit more complicated as you go later on, but it's the method of separation and it depends on the equilibrium between the mobile phase and the stationary phase. Now, the stationary phase at this stage is going to be paper. So at GCSE, it's going to be paper, right? It's not going to be anything else. Mobile phase is going to be water generally, but it can be any solvent, but it's usually water at GCSE. And all we're going to do is try and separate out different dyes using this. Now, how do we do that? We do that in the lab, okay? And we do that using a piece of paper. So here's my piece of paper. Okay, and that's my stationary phase, the bit that doesn't move. Okay, then we draw a baseline on it. Now that baseline is done in pencil. The reason is you're going to put solvent all the way through this. So if this is anything but pencil, it will dissolve in the water and go with all your dyes that you put on. So really important, and that's an exam question. Why is the baseline in pencil? The baseline is in pencil because it doesn't dissolve in the solvent or the water. We place then our dyes onto our baseline. We usually do that with a spotter. Okay, and there's 3 dyes that we've chosen for today, dye A, dye B, and dye C. We then place it in a beaker, and then we put solvent in the beaker. Sometimes if you're in the lab, you'll have the solvent in the beaker first, and then just put the paper in. Now, often as well, we can hang the paper on something just to keep it upright, because we need to keep it really upright. We then put the lid on top. And then what happens here, because the lid is on top, the water can't escape. So the water just evaporates a little bit, fills the space here, but then actually the water just goes up the paper. Okay, if we didn't have the lid on, that wouldn't happen. All that would happen is the water would just evaporate and it wouldn't work. Okay, so we need that lid on. Now, if you then look at that, we've got different colours of dyes from our previous sample. So I'm going to go back and just show you. Remember, we had 3 different samples, and then we ran the chromatogram, and the water goes up. Okay, and now we've got all of these different dyes in these different samples. So really important in the first one, we have a yellow dye and we have a red dye. But you can see, because it's on the same level, you know that these are the same dyes or the same colours or the same compounds, okay, because they're on the same level as they go up. And we also have a blue dye and a black dye. So things to observe. Now, what I'd like you to do is stop the video and just tell me what you observe from this. So what you observe is this. This sample, which was A, has 2 different compounds, 2 different dyes. This is B, it has 2 different dyes. And this is C, it only has 1 dye. So that's your first observation. Okay, then this is A, this is C. Okay, A and C have the same dye in them, both have red dyes, and the rest are different. So you can get these kind of observations from here, and they're common exam questions. Very simple, but you need to get used to doing them. And remember that baseline's in pencil, so it doesn't dissolve in the water as the water goes up. Now, if we then put that to the side and just summarise this, the distance travelled by the dye is changed by the attraction for the mobile phase, which is the water, so the water is going up, and the stationary phase, which is the paper that it is on. So if it's more attracted to the stationary phase, it's going to stick to the paper, and if it's more attracted to the mobile phase, it's going to move up the paper. But we'll show you that in a diagram in a second. The same line, height, means the same compound or dye, and the number of dots equals the number of compounds or dyes in the sample. Now, all of this can be measured in terms of something called an RF, and we'll go over RF in more detail later on in this video, but RF is just how far the spot or the dye has gone up the paper, and that is relative to the solvent front. So I haven't mentioned the solvent front yet, but the solvent front is just how far the solvent got up the paper. So if you're doing this in the lab, you put it into a beaker, you'll let the water go up, and then you'll see where it got to, and then just draw a line in pencil again, because you don't want the pencil to dissolve in the water. Okay, So you can see here that this is about halfway up. So its retention, its RF would be about 0.5 because it's 0.5 up the paper. This is about 0.85 up the paper. That's about 0.8 up the paper. So it's that kind of thing. But we'll show you how to do that mathematically later on in the video. So let's have a look at this attraction for the paper. We're going to go show you an exam answer here that you really do need to have in your notes and have it really important. So here's our two samples. We've got a blue sample and we've got a yellow sample. We're going to flip these so they are horizontal. So we flip them so they're horizontal. And hopefully you'll recognise here this little molecule right here. That's a molecule of water. So it's just a diagram of a molecule of water. Now we know as the chromatography happens that the water goes up the paper. So it's going to go in that direction, it's going to go up the paper. Okay, and if we then make the water do this, okay, and there the water goes up the paper. So the water's gone up the paper, but you can see on this one, the sample has only gone, I don't know, to just under halfway up the paper. This one's gone all the way up the paper, right? So that's gone all the way up the paper. Now, you need to describe this in the exam, whether this is an attraction for the paper or an attraction to the water. And how do you write that out? So looking at this one, this has only got partially the way up the piece of paper, okay, which means that it must be more attracted to the paper. So it's stuck on the paper and the water is not really dragging it along. Now, in terms of an exam answer, here's your exam answer and your exam marks. This is 1, 2, 3, okay? So the compound or the dye is attracted more to the paper. That's our little lines here, okay, and spends more time attracted to the paper, okay? That's relative to the solvent. So it doesn't spend more time in the solvent, it spends more time being attracted to the paper. This RF value is just how far it goes up, and it's not gone very far, so it has a low RF value. So there's your 3 marks. Now what I want you to do is stop the video and try and write down the same description, but for this one. Okay. So the next one, more attraction to the water, because the water is dragging it up that paper. Okay, so the compound dye is attracted more to the solvent. The other one was more to the paper. This is more to the solvent. Okay, so there's more time dissolved in the solvent, and therefore has a higher RF value because it's gone further up the paper. And you can see that now if I just rearrange this all. Okay, and you can see that's now rearranged. You can see this is way up the paper. I've now put a solvent front in just to make it really clear as well. So that means it's more attracted to the solvent, spends more time attracted to the solvent on this one, and on this one, it's more time attracted to the paper because it's further down. And that is the exam answer when it comes to chromatography and describing why the dyes go up the paper and have a high RF or stay further down the paper and have a low RF. The next part is how to do it in the lab, and this will be an exam style question as well. So quite long and drawn out, okay, but these are the instructions. So everything in orange though is going to get you a mark, and everything in white is just the little fine details, okay, for you to do in the actual experiment. So draw that pencil line. Remember that pencil line must be in pencil because it doesn't dissolve in the solvent. And then you take a capillary tube, small tube, okay, and just drops on the baseline. Put your samples on the baseline. Repeat to try and make it concentrated, but not make the spot large, otherwise they run into each other. It's not very good. Do the same with other dyes, making sure they're separate from each other. And place the paper in the beaker. Remember there's solvent already in the beaker, and that solvent must not go above the baseline because you don't want it to dissolve your dyes. Okay, now place a lid on top, really important, and allow the solvent to rise up the paper close to the top. Take out and draw a line, that's your solvent front, and again do it in pencil so it doesn't dissolve, okay, and leave it to dry. And then you'll usually sellotape that into your book if you've got one, or if you're working on tablets, you take a picture and put it into your thing like OneNote or your note program, okay. But that looks really long, but it's not, okay. So what's going to get you marks? Let's just do this. Okay, and you can see, I've just taken out all the bits that are really important that are going to get you marks. Ding, 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 ding. Okay, these bits are going to get you marks. So if you're going to do your notes, make sure you write down these. Make sure that these are perfect. So we're now going to go into more detail on retention factor, or RF. Now this is a simple calculation that you have to do to see how far it's gone up the paper. Okay, so if you can work out how to do this already, please do. Okay, and make some examples for yourself. So draw out some chromatography papers, put some spots on and work out the RFs of the different spots. Now, the RF reflects the distance of the compound up the paper relative to the solvent front. So it started at the baseline and it went up the paper. So it's how far it's gone compared to the solvent front. And you use it for identification. So same RF, same compound, same dye. Based upon the balance between the attraction for the water, for example, if it's more attracted to the water, it's going to be up here. If it's more attracted to the paper, it's going to be somewhere down here. Okay, so it's that balance of attraction and how much time it spends being attracted to the water and how much time it spends being attracted to the paper. Now, if we then have a look at this and put a ruler on. So we've got a ruler on, you can read these off if you like, stop the video and read it, make it a bit more interactive. So we've got here, we're just measuring the distance. So we're going to measure the distance of each die, and we're going to go to the center of the die. So that's what we do, go to the center of the die. So if there's a spot, go to the center of the spot. Now you can see my arrow right there, and then I can measure that, and that's 27 millimeters. We then go back, and we measure the red dot, okay, and that is going to be 48 millimeters. So we've got our millimeters that we're calculating here. Okay, and now we're going to do our solvent fronts. We're going to look at our solvent front, see how far the solvent front is, because the RF is the distance moved by the compound. Okay, as we can see, 27, 48, and I've filled in the other ones, 43 and 32 for the blue and the black. Okay, but we need to know the distance moved by the solvent, and that is the distance between the baseline and the solvent front, which is 60 millimeters. All right, so you can now stop the video and you can do the RFs of all of these if you like. So here are all the RFs. So we've got our yellow dye, just take the distance it's travelled by the distance moved by the solvent, which is the same each time. Okay, and you get a number between 0 and 1. And the higher the number, the further up it has gone up the paper, the more time it has spent in the solvent and being attracted to the solvent. So that means the red dye is attracted to the solvent more than it is attracted to the paper and spent more time being attracted to the solvent. Whereas if we go to this one, we can say the yellow dye is more attracted to the paper, spent more time being attracted to the paper, and therefore hasn't moved very far up the paper and has a lower RF. And that is how we do retention factors, and we can relate them. But these calculations are very common in exams, and you should definitely practice them.